Hello everyone, this is Jerry Ellison Mutiabele and I'm going to be taking you through receptors and pharmacodynamics. Remember, when we talk about pharmacodynamics, we are looking at what the drug does to the body. So let's start by defining what a receptor is. A receptor is simply a binding site in biological tissues for exogenous or endogenous ligands. And what is a ligand? That is simply a molecule that binds to a receptor. So a receptor can also be protein molecules in the cell where specific ligands may bind. Simply a protein molecule in the cell where specific ligand may bind. So we are going to look at the different receptor families. But first of all, receptors are embedded in, we can find our receptors embedded in cell membranes, and those are called cell surface receptors or transmembrane receptors. We can find them embedded in the nucleus. We call those ones nuclear receptors. So the cell surface receptors belong to three major families. These include the G protein coupled receptors, the ligand gated ion channel receptors, enzyme linked receptors. So let's start by looking at the G protein coupled receptors. We know that this is the most abundant type of the cell surface receptors. So under this G protein coupled receptor, we have a receptor being coupled to a signal transducing guanine protein or a G protein. And ligand binding, when a ligand in this case, we have a drug molecule, binds to the receptor, it activates the G protein coupled receptor. So the activated G protein then activates cellular enzymes such as adenylyl cyclase or phospholipase C. So if it is adenylyl cyclase that is activated, it will go on to act on ATP, generating cyclic AMP. Then if it is phospholipase C that is activated, it will act on phosphatidyl inositol to generate inositol triphosphate and diacyl glycerol. So the cyclic AMP, diacyl glycerol, and inositol triphosphate are simply referred to as secondary messengers. So cyclic AMP phosphorylates or activates a cellular kinase, in this case protein kinase A. So this cyclic AMP will phosphorylate protein kinase A, in that case activating it. Then diacyl glycerol activates protein kinase C, which is responsible for regulating proteins with catalytic activities, bringing about cellular responses. Then we have inositol triphosphate eliciting calcium ion released from the endoplasmic reticulum. Then this calcium activates calmodulins, which are simply calcium binding messenger proteins, and calcium dependent kinases. This will result in increased cell metabolism, smooth muscle contraction, and other cellular responses. So we have inositol triphosphate eliciting calcium to release from the endoplasmic reticulum. Then the calcium that is released will then go on to activate calmodulins and calcium dependent kinases. All these will result in increased cell metabolism, smooth muscle contraction, and other cellular responses. So if the ligand binds to the G protein coupled receptor with a G stimulatory protein, that's a G stimulatory protein, the cascade is activated, leading to a response, and vice versa for G inhibitory proteins. So that means when the ligand binds to the G protein coupled receptor with a G inhibitory protein, the cascade will be inhibited. So our secondary messenger, in this case cyclic AMP, is broken down by phosphodiesterase enzyme, that is to regulate the reaction. So when the work of cyclic AMP is done, it is then broken down by the phosphodiesterase enzyme. Now we know that phosphodiesterase inhibitors, in that case, will enhance the response. Meaning, when we have these inhibitors, they inhibit the enzyme. That means the enzyme will not be able to break down cyclic AMP, and therefore we shall have more cellular response. Examples of these inhibitors, we have xanthines and milrinone. And remember, as we're talking examples of xanthines and pharmacology, we have things like caffeine, so this is the process. We have our ligand. In this case, it will be norepinephrine, the neurotransmitter, binding to our G protein coupled receptor. In this case, it is the beta adrenergic receptor. So it will be activated and it will go on to act on ATP, generating the secondary messenger, which is cyclic AMP. So the secondary messenger cyclic AMP will go on to phosphorylate or activate protein kinase A. Now the activated or phosphorylated protein kinase A will now go on to bring about cellular responses. So let's look at an example where we have phospholipase C as our enzyme. So we have our ligand, in this case angiotensin 2, binding to the G coupled receptor, which is the angiotensin receptor, activating it, and it will go on to activate phospholipase C. The activated phospholipase C will then act on PIP, generating inositol triphosphate and diacyl glycerol. So we said the diacyl glycerol will go on to activate protein kinase C, bringing about cellular responses. Now, inositol triphosphate will cause calcium release from the endoplasmic reticulum, and this calcium will be the one that will bring about our response by acting on calmodulins, bring about cellular response. So let's look at examples of receptor subtypes under this family of the G protein coupled receptors. So here we have a receptor type and the ligand that binds to that specific receptor. So for beta adrenergic, the beta adrenergic receptors, we have epinephrine, dopamine, and norepinephrine as our ligands. Then for histamine receptor, we have histamine H1 and H2, and our ligand will be histamine. For prostaglandin receptor, we shall have our ligand being prostaglandin. For the serotonin, that is 5-HT, we shall have our ligand as serotonin. Then angiotensin, as we saw, angiotensin 2, our ligand will be angiotensin. That's the ligand will be angiotensin 2 for the angiotensin receptor. Then for the muscarinic receptors, we have muscarinic 1 and muscarinic 3, and our ligand will be acetylcholine. So let's go on to the ligand-gated ion channel receptors. So ligand binding under this changes conformation of the receptor and causes opening of ion channels. So under ligand-gated ion channel receptors, when our exogenous ligand binds to the receptor, it will cause conformational change of the receptor or morphological change of the receptor, leading to opening of ion channels. So ions such as sodium, potassium, chloride, movement alters the electric potential across the membrane, leading to depolarization and anaction potential. That is when acetylcholine causes skeletal muscle contraction. Then we can have hyperpolarization when acetylcholine causes a decrease in heart rate. So the activation of these ligand-gated ion channels may be direct or G-protein linked, and these receptors are found in nerve and muscle cells. So let's look at a ligand-gated ion channel receptor here. We have our ligand, we have the ligand binding site, and then the receptor protein. So when a ligand binds, there will be conformational change leading to the opening of these channels, allowing exchange of ions So now this exchange of ions can either lead to depolarization or hyperpolarization. Now let's look at the directly activated ligand gated channels. Remember, we said we have directly gated ligand gated ion channels, and then we have the G protein linked gated ion channels. So let's look at the direct ones. They are simply called direct because when our ligand, in this case we are taking the example of acetylcholine, binding to the receptor, in this case nicotinic 2 receptor, which is a cholinergic receptor found at the neuromuscular junction. When acetylcholine binds to the nicotinic 2 receptor, it will lead to conformational change, hence opening of the ion channels. You can see the ion channels opening, and this will lead to the influx of sodium ions into the cell. So the influx of sodium ions into the cell will lead to depolarization, hence generation of an action potential, leading to muscle contraction. So now let's look at the G protein linked ion gated channel. In this case, we are going to take an example of acetylcholine still. So when acetylcholine binds to our receptor, which is linked to the G protein, in this case we have a G inhibitory protein like we saw. Now we know what it does, and in this case we are looking at a muscarinic 2 receptor that is found in the heart, particularly in the sinoatrial node. That's where our receptor is found. And when acetylcholine binds to this receptor, which is G-linked, the alpha unit will break off. Then the beta and gamma unit will go and bind to the ion channel, causing it to open, leading to exchange of ions. So there will be efflux of potassium ions and influx of chloride ions. So this will cause the negative charge in the cell to be higher than the resting membrane potential, hence leading to hyperpolarization. So this hyperpolarization is like an inhibitory response. So in this case, we shall have a decrease in heart rate. So that means when acetylcholine binds to the muscarinic 2 receptor, it will lead to a decrease in heart rate. So examples of receptor subtypes under this family also. the ligand-gated ion channel receptors. We have the nicotinic 2 receptor, where acetylcholine is going to act like we saw. We have the muscarinic 2 receptors in the heart, where still acetylcholine is going to act. Then we have N-methyl D-aspartate receptor, where we have our ligands such as aspartate and glutamate, which are amino acid ligands. Then we have the GABA receptor, where the GABA ligand binds. Glycine receptor in the spinal cord, where glycine binds. So let's go to the third family, which is the enzyme linked receptors. So these receptors are linked to enzymes that don't have intrinsic catalytic activity. An example of these enzymes is tyrosine kinase. So when a ligand binds to the receptor, it will activate tyrosine kinase, which is actually linked to the receptor. So the activated tyrosine kinase transfers a phosphate molecule from ATP to cellular kinases, which then will become phosphorylated because they are receiving a phosphate molecule from ATP being transferred by tyrosine kinases. So the cellular kinases then activate signal transduction pathways, leading to increased mitosis and therefore cell proliferation. So when our cellular kinases are activated by being phosphorylated, they will also activate signal transduction pathways, hence leading to increased mitosis and cell proliferation. So we can see here an illustration when the ligand binds to the receptor that is linked to tyrosine kinase. The tyrosine kinase will be activated and it will transfer a phosphate group from ATP to the cellular kinases. So now the activated cellular kinases will then activate the signal transducing pathways, leading to mitosis and proliferation. So, what are the examples of ligands that are going to bind to these kinds of receptors? We have cytokines, such as interleukins and interferons, and we know cytokines as signaling molecules in the body. So let's say during immune response, when these cytokines bind to the enzyme linked receptors, they will activate macrophages and T lymphocytes. So remember when we said the signal transduction leads to mitosis and proliferation. So in this case, we shall produce macrophages and T cells. Then we have erythropoietin, which is involved in red blood cell production. Remember, under enzyme linked receptors, we are looking at increasing mitosis and proliferation. So when erythropoietin, which is our ligand in this case, binds to its receptor, we shall have increased RBC production. Another one is growth factors, when they bind to their specific receptors, they will cause epidermal growth factor release or production. So now we've finished the cell surface receptors. Now let's go on to look at the nuclear hormone receptors. These receptors are intracellular and mainly for hydrophobic ligands, that is, water-hating ligands or lipid-loving ligands. So meaning these ligands are able to pass through the lipid bilayer of cell membranes. Unlike our previous ligands that we saw binding to receptors on the cell surface. So the receptor ligand complex binds specific DNA sequences. And what is the receptor ligand complex? We shall see. In this case, our receptor, in this case, the nuclear hormone receptor has a ligand binding domain, meaning the area where the ligand is going to bind, and also has a DNA binding domain, meaning the area that is going to bind to a specific DNA sequence. So we are looking at our receptor having two domains. One domain where our ligand coming from outside is going to bind, then another area where it's going to bind to a specific sequence of DNA. So the specific nucleotide sequences of DNA to which the DNA binding domain binds are called hormone response elements. The hormone response elements are just those specific sequences in the DNA to which the DNA binding domain is going to bind to. Remember, we said the DNA binding domain is part of the receptor because it's made of a DNA binding domain and the ligand binding domain. So what are the examples of ligands for these receptors? Remember, we are looking at ligands that are hydrophobic. So we have steroid hormones such as estrogen, testosterone, progesterone, and thyroid hormone. Another one, we have the glucocorticoids, that is cortisol and dexamethasone. Then fat-soluble vitamins such as A and D. These are the ligands for these nuclear hormone receptors. They are hydrophobic, and we have the steroid hormones, glucocorticoids, and fat-soluble vitamins. So they regulate metabolism, growth, and reproduction. That is, they lead to increased muscle mass. So when these ligands bind to the receptors, they will lead to regulation of metabolism, growth, and reproduction. And their effects are long-term. That is, follow gene expression. Because remember, gene expression, it's a long-term thing or long-term response. So the effects tend to be prolonged also. Now effects like growth, it's a long-term thing. So we have an illustration here. We have our ligand. It can be a glucocorticoid, a steroid hormone, or a fat-soluble vitamin traversing the cell membrane. Because it is hydrophobic, it can pass through the lipid bilayer easily and enters the cytoplasm. So while in the cytoplasm, remember we said our receptors in this case have a ligand binding domain, and they will also have a DNA binding domain. So that simply means an area where the ligand is going to bind and an area where it's going to bind to the specific DNA sequence. So here we can see our steroid hormone, which is the ligand binding to the ligand binding domain, forming the hormone receptor complex, like we mentioned. So this will then pass through the nuclear pore into the nucleus, and when in the nucleus, this complex will bind to receptor sites on chromatin of DNA, activating messenger RNA transcription. So our complex will bind to a specific sequence of DNA, leading to activation of messenger RNA transcription. And with transcription, you know that what follows is translation. So let's look at some of the properties of receptors in general. One of them is that these receptors are specific. That is, there is a structural complementarity to the ligand. So a specific receptor is specific to a given ligand structurally. So the ligand will have its structural configuration that is specific to bind to a specific receptor. And they're telling us that drugs with similar structure tend to bind to the same receptor because they have the same structural configuration. Another property is saturability, where the number of receptors per cell is finite, meaning it's not endless. It can be finished or filled up. And at saturation, maximum response is expected. When all the receptors have been filled up or saturated, which we are calling saturability, we expect maximum response. Another property is reversibility. That is, being able to be reversed or brought back to the beginning or the origin. So a ligand should bind to the receptor and then dissociate. So its action is not indefinite. So this is useful because imagine these drugs were actually binding to receptors and they will bring about a response, and that response was prolonged. Yes, that would not be very good because actually drugs are just supposed to come in to modify body processes, and when they are done, they are supposed to dissociate and be degraded. They are not supposed to bind irreversibly. So another property of these receptors is reversibility. Thank you for listening.
Hello everyone, Kazwe Patrick once again, and today I'm going to discuss the dose-response relationship. Now let us understand what dose-response relationship is all about. Simply, it is an interaction between the dose that is administered and then the response that is produced. And it is based on the occupation theory, receptor occupation theory, which states that the effect of the drug is directly proportional to the fraction of the receptor the drug occupies. For instance, for a maximum response, all receptors are bound by the drug. So the dose-response relationships have what we can call as the son or the daughter, and this is the dose-response curve. So the dose-response curve is a plot of the percentage of the response against the concentration of the drug, and it is the basic currency of receptor pharmacology. Why we call it the best currency is because it is used in many areas as we are studying the receptor pharmacology. For instance, in comparing of the drugs, in studying of the therapeutic index and safety, in studying of the potency and the maximal effect. So in many areas of pharmacology, it is used. And then there are two forms of the drug response curve. So we have the dose response curve. We have the one which is hyperbolic and then the one which is sigmoid. The one which is hyperbolic is whereby the agonist concentration is given on a linear scale, and then the other which is sigmoid is one whereby the agonist concentration has been converted to a log scale. So why there is a conversion from the linear scale to the log scale is because that you find that some concentrations are much smaller while others are much larger, and so we might fail to have a better visualization and interpretation of our results. So it's better when we can convert them to the log scale because it provides a better ground where we can easily interpret and analyze our data. So the following are some of the keynotes that we need to put at the back of our mind as we are interacting with this drug response relationship, the dose response relationship, and then the dose response curve. One is the Emax, and it is indicated properly on the curves in the previous slide. So the Emax is the maximum effective dose, which is the dose or concentration of the agonist that is producing maximum effect. Then the EC50 is the effective concentration for 50% response, and this is the concentration or dose of the agonist that is producing half the maximal response. Another note is that the drugs do two things to receptors. They will either bind to them, and this binding will be based on the affinity, and as well when they bind they will change the behavior of the receptors, and this will be based on the efficacy. The following are some of the important terms and concepts that we need to know as we are studying the dose-response relationships. One is affinity. Affinity is simply the ability of a ligand to bind to a receptor. Then efficacy is the ability of the drug receptor complex to produce a maximal response. And the agonist is a ligand or a drug that binds to a receptor and triggers a pharmacological effect. Then the inverse agonist is the ligand that binds to the same receptor as the agonist but induces an opposite pharmacological response to that of the agonist. The partial agonists, they fail to elicit maximum response even when all receptors are occupied. So for them, they have low efficacy. As we show initially that efficacy is the ability of the drug receptor complex to produce the desirable or maximum response. So for the partial agonists, for them, they will have a low efficacy. They will not have that ability to bring about the maximal response. An example is the naloxone for opioid receptors, meaning that even when we have naloxone in high concentration and all receptors have been bound to, we can never have a maximum response. Then a full agonist, for it is a ligand that elicits a maximum response after occupying all or a fraction of receptors. Then sparse receptors are a fraction of receptors that have been left unoccupied after full agonist binding. So in this slide, we are going to first appreciate the log dose response curve, which is LDR, for the full agonist and partial agonist. So we have seen that for full agonists, for them, they will be able to produce a maximum response when all or part of the receptors have been bound. Then the partial agonists, for them, even when we have all receptors bound, they will not be able to produce a maximum response. As we can see, A and B, they have been able to produce a maximum response, though at varying concentrations. This might be due to other factors like presence of competitive inhibitors. But for C, it has failed even when there are no inhibitors. Then on the left here, we can see some of the examples of the full agonist. So we have the receptors here and then the examples. One is where we have the opioid receptor, whereby the full agonist is morphine. Then dopamine receptor, the full agonist is dopamine. GABA, the full agonist is thiopental, and many of these others. Now, antagonists, for them, they will bind to receptors, but they will produce no response in absence of an agonist. In simple terms, they have no efficacy. Their interaction with the receptor will produce no response. So we have two types, the competitive antagonist and then the non-competitive antagonists. So the competitive antagonists, for them, the antagonist has the same receptor binding site as the agonist, and its presence will decrease the agonist's response, and it will make the drug or the dose response curve shift to the right. So the antagonist decreases the agonist's affinity but not efficacy. Therefore, an increase in the agonist concentration can produce a maximum effect or response. In this slide, we are going to appreciate the effect of competitive antagonism. Yes, so in the first curve where we have antagonism alone, you can see that the maximal effect or response is attained at lower concentrations of the agonist. Then in the second curve where we have the antagonist, the competitive antagonist, we see that also we achieve the maximum response but at higher concentrations of the dose of the agonist. So this simply means that the antagonist is affecting the affinity of the agonist to the receptors but not the efficacy. And in this other side, you can appreciate the differences between the agonist and the antagonist, where we are seeing that the agonist will bind to the receptor and there is a pharmacological effect or a response. While for the antagonist, it binds to the receptor and we shall have no response in case of absence of the agonist. Then here where there is an agonist, there is an effect, but the effect is lessened. So for the non-competitive antagonist, for them, they prevent the conformational change that triggers the response. So the antagonist binds to the allosteric site. So this is a site that is different from the agonist binding site. and it will lead to a conformational change in the receptor, whereby now the receptor will no longer be fitting the agonist. So the effect here is not overcome by increasing the agonist concentration, because the antagonists, they produce non-surmountable antagonism. So this is simply the antagonism that cannot be overcome. So in this slide, we can appreciate the effects of the non-competitive antagonists, whereby they are bringing about the non-surmountable antagonism which cannot be overcome. We see that even though we increase the concentration of our agonist, the maximal response still is below the expected one without the antagonist. And this antagonist, we see it will affect both the efficacy and the affinity, because it binds to the allosteric site and to bring about a conformational change, whereby our agonist is denied the ability to bind to the receptor, meaning its affinity to bind has been inhibited. As well, we shall not have the interaction between the receptor to bring about the desired response. So as we have been introduced to the dose-response relationship, there are two forms of the dose-response relationship, and the first one is the graded dose-response relationship. So this one is studied in vitro. So this is an isolated tissue that has been placed in tubes within the laboratories, and with it the magnitude of the response is measurable. For instance, when we increase the dose, there is an increase in the amplitude of contraction. So as we can see here for this example of carbaco, so as we are increasing the dose of carbaco, the amplitude of contraction also increases. This graded dose response relationship is used in efficacy studies that will be looking at the maximum effect with the dose. So the example is in this table, where we have the effect of cholinergic agonist, that is carbaco, on contractility of the isolated gut ileum in an organ bath that has been attached to a transducer that is connected to a chymograph drum of the paper. So this chymograph drum is the one which will be giving us the amplitudes. That is the distance from the starting line as the wave goes up. It is this amplitude, and it is the one that will be measured as the response. And as we are increasing the dose, the response or the amplitude also increases. The second type of the dose-response relationship is the quantum dose-response relationship, and for it is studied in vivo, that is in live animals, and it is all or none response, whereby as we are studying our population, either the individuals within this population, they will produce the response, or there will be no response. Then the measure of the response is equal to the percentage of the animals responding. In this case, the number of dead animals. And this quantum dose response relationship is used in the safety or toxicity studies, because it allows us to identify the dose range where the adverse effects occur, and we are able to establish the appropriate dosing guidelines, as we can see in this table. So as the drug dose is increasing, we have a constant number of animals that we are studying on. So as the drug dose is increasing, we shall show that the response, which is the percentage of death of the animals, also is increasing. And at this concentration, which is 8,000 milligrams per kilograms, we are having 50% of the population succumbing to death. And this concentration will be called the median lethal dose, LD50, which is the dose at which 50% of the population exhibits the specified one-to effect. And in this case, what we are looking at as death. Thank you very much for listening and watching. See you later.
Hello everyone, I'm Kato Lawrence Kanyike. Today we are going to look at the potency of drugs and their therapeutic index. So potency of a drug is basically the dose that must be administered to produce a particular effect of a given intensity, or it's a measure of the amount of the drug required to obtain a particular response. Well, potency varies inversely with the dose, so they are inversely proportional. It is influenced by the drug's affinity for its receptors. In other words, the higher the affinity of the drug for its receptors, the higher its potency. So it's also affected by the pharmacokinetic processes such as absorption, distribution, and elimination. So here the key points are that potency of a drug is basically the drug that is administered to achieve a particular response of a given intensity. So here we have the prerequisites that must be considered before comparing drugs for their potency. The first one is that these drugs must have the same mechanism of action and the same pharmacological effect. For example, here we have the barbiturates such as thiopental, thiobarbital, and secobarbital. These are barbiturates and they are all GABA receptor agonists and they are sedatives, so they have the same mechanism of action and the same pharmacological effect. Another point, they should be administered using the same route. You either use subcutaneous for all of them, you can use IV for all of them, or the PO, Parose oral route for all of them. It should also be administered to the same species of animals. So if you are administering all of them, they should be administered to the same animals, such as a guinea pig or the rats in the laboratory, such that we have the same comparison for all these drugs. So in this slide, we are comparing the potency of the neuromuscular blockers, that is galamine, atracurium, and oxacurium. The drug with the highest potency. For a drug with the highest potency, we shall require a lesser dose of that drug in order to achieve the required intensity. And then followed by this one in the middle, it shall have a median potency to require that same intensity, and this with the low potency we shall require a higher dose to achieve that particular intensity. So here we are comparing the potencies of those drugs. So here we are looking at the safety of drugs, and this is evaluated by relationship between the doses that produce the desired effect and those that produce the adverse effects. And these are determined using the quantum dose response relationships. This is determining in vivo, in contrast to the graded dose response relationships, which is done in vitro. Here the adverse effects that we have are serious or life-threatening, and they may require reduction of the dose or cessation of the therapy. For example, for aplastic anemia, where you have prolonged use of the chloramphenicol therapy, you have more susceptibility to aplastic anemia. Then teratogenicity, if you overuse thalidomide therapy, and also for heart attacks after rofecoxib or Vioxx. And the side effects here, you rarely require reduction of the dose or cessation of a therapy in case of toxicity. So you might require to reduce the dose, or you might require to cease that therapy in order to avoid toxicity. So we proceed to therapeutic index of drugs. Well, this is based on the safety of the drugs as explained in the previous slide. It is the quantitative measurement of the relative safety of the drug. In other words, we compare the dose that produces the desired effects with that produces the adverse effects, as I explained in the previous slide, and is given by the formula medium lethal dose, which is LD50, and the median effective dose, which is ED50. In other words, The median lethal dose, or LD50, is that dose that kills 50% of the test population, and the median effective dose, or ED50, is the dose that produces the desired therapeutic effect in 50% of the population. In other words, if I inject 10 rats with chloramphenicol or carbaco, and I test the number of rats that will die, 50% of the rats that die, that means that's the dose that will produce 50% of deaths for the rats. That will be the median lethal dose, and the dose that will produce 50% of the desired effect in those rats injected with carbaco, that will be the median effective dose. So here, that's how we go about the therapeutic index of the drugs. So here we are trying to appreciate the concept of therapeutic index. Here in this graph, the blue line, we are having the effective dose, the desired effect, and here in the red graph, we are having the lethal dose, or the undesired effect. So here for the required dose, the ED50, which is the median effective dose, is 8 at this point. You see it corresponds with 8 here. And then here the median toxic dose, or the median lethal dose, which is 80 here. So this is the safety margin. The difference between the two is the safety margin. If we do the math and we substitute into our formula of therapeutic index, the median lethal dose, or the median toxic dose, which is 80 from here, and then the median effective dose, which is 8, if you do the math, you come up with 10 as the therapeutic index. Well, another concept that you should take note of is that the higher the therapeutic index, the lower the dose, because then you will require a higher dose to reach the lethal dose, the lethal point, and the lower the margin of safety, or the therapeutic index, then the less safe the drug is. So for those drugs, you have to take caution. So this is the whole concept of therapeutic index. In this video, we looked at the potency of drugs and the therapeutic index. Thank you for watching the Innovates YouTube channel.